I'm taking Coral Warrior. That's my self-proclaimed nickname. I love it. I'll find one for you too, Megan. Thank you everybody for having us here today. I'm Steve Vollmer, Director of Florida Atlantic's School for Environmental, Coastal, and Ocean Sustainability. We're a relatively new school at Florida Atlantic. There's lots of representation from Florida Atlantic in the audience. I was proud to see that. Can I see a quick show of hands of anybody that's graduated from Florida Atlantic? Awesome. Before I begin talking about my talk, I'd also like to acknowledge our 10 undergraduates. You guys want to stand up? These could be your future interns, your future employees, your future eco-warriors, I would say. And as Megan said, she asked me to tell you what the state of the oceans were. She took my punchline, and I was going to say, I'm not going to cover 70% of the Earth's surface in 25 minutes. And I was going to stick closer to the chest and talk about what I love, which is coral reefs. And so I'm giving you my perspective as a coral biologist for the last 25 years. Where I started my journey working in coral ecosystems as a PhD student at Harvard, worked in Panama at the Smithsonian Tropical Research Institute, spent many years in Boston at Northeastern, and now proudly joined Florida Atlantic University to continue to be a coral warrior. And when you think about coral reef ecosystems, sometimes this is what comes to your mind. This is a beautiful, healthy reef in the center of the Coral Triangle in Indonesia. And you can see why they often call coral reefs the rainforests of the oceans. I like to think of rainforests as the coral reefs of the land, because coral reefs are actually the most hyperdiverse ecosystems that we have on our planet. There's 100 corals of species sitting in front of us in our plain view, and hidden within all the cracks and crevices are myriad thousands to millions of species of animals, plants, algae, sponges, and a bunch of microbes. They're just teeming with life. But I think we also need to think about our coral reefs as resources. So our coral reefs fuel our economies. They're providing hundreds of billions of dollars to our global economy, and they're doing it across nations across the globe. But like our oceans, we often see reefs from the surface, not below, and we extract from them. Our reefs are not infinite, our oceans are not infinite, and yet we often have been extracting without putting back into them as the resources that they are. They provide protection to our homes and our coasts. You guys have been talking about that a lot. They're putting the food on our plate. They're the source of our new medicines, our new cancer drugs, probably the wrinkle cream if you put it on your face. That's coming from a coral reef, actually a coral. And so we need to start thinking about putting back into these ecosystems. And Florida's reef-based economy alone provides $8.6 billion and supports 70,000 jobs to our local economy. And we need to invest in them like the resource that they are. And as many of you know, coral biologists like me have really raised the alarm of the threats to our coral reefs. So we often see more and more the phenomenon called mass bleaching. That's what you're looking at right here. And it's rising seawater temperatures that Obe showed us yesterday. The oceans are basically absorbing our heat that we're producing at a faster rate than any other part of the globe. And the reefs are kind of the canary in the coal mines to show the dramatic effects of the heat that we're sinking into our oceans. So what you're looking at here are corals that are basically dying. Their normal color is brown, and that's because of the classic symbiotic relationship between the coral animal and its algal host. When corals get stressed out by heat, that breaks this classic bond, and the coral literally digests or expels its algae and starts starving to death. They get 90% of their food from the relationship. Every one of these pictures of the corals that you're seeing here, which is taken on the Great Barrier Reef over the 2016 bleaching event, all the white ones, even the very beautiful purple ones, they're actually going to die. And they're not going to recover. Every one of these coral colonies is greater than 10 years old, so it takes a long time to build back our reefs. Now, for a long time, we thought our reefs in the Great Barrier Reef or in far-flung locations like Fiji or Indonesia, we thought they were immune from bleaching because they were so resilient or they were so remote. But since the Great Barrier Reef bleaching event in 2015, we've seen bleaching events affecting the reefs across our globe, and none of our reefs are immune from the rising sea surface temperatures that we're imposing on our climate and on our reefs. But if you work in the Caribbean or you work on Florida reefs, we've seen the impacts of our own inputs on the reefs, negative inputs on our reefs, happening for a long time. So this is a picture provided to me by Ken Niedermeyer from Reef Renewal, a classic pioneer that you'll see in a minute, of Carysforth Reef in 1974. So this is Upper Keys. And these types of pictures were commonplace throughout the Caribbean. This is our now endangered elkhorn corals, a beautiful thicket covering the front of Carysforth Reef. These were all over the Keys, all over the Caribbean. Down below them, you would have seen staghorn corals, also endangered. And now below them, you'd see your beautiful brain corals and your boulder corals. But our reefs in the Caribbean and the Keys have been declining for decades. This is a report that kind of shows what we've known all along. This is a report that came out last week, reported in The Guardian, but published elsewhere, where we've basically lost 50-ish% of our hard coral cover since the 1980s. Okay? Those are actual numbers, but you can see these transitions in these pictures, just plainly. So Gene Shin, an old coral biologist and geologist, in 1971, he was taking pictures of our beautiful reefs. The top one is Grecian Rocks. The bottom one is Carysforth Reef. This is basically a classic Caribbean reef in the Keys. Elkhorn coral in the shallows, staghorn coral underneath, right at the reef crest, boulder corals deeper down. By 1988, the staghorn and the elkhorn corals have died due to white band disease, the first major heavy impact on our reefs. And then by later on in 1988 or 98 and 2015, even this massive boulder coral has succumbed. And these types of phase shifts have happened throughout the Caribbean and throughout the Keys. We're just losing our corals at an alarming rate. And there's three reasons why, primarily. So the biggest three reasons why we've lost our corals in the Caribbean and Florida reefs are three factors. First was the devastating impacts of white band disease starting in 1979. That would be off the scale here. Second is a new epidemic of disease called stony coral tissue loss disease, which emerged in Miami and has spread through Florida. We'll talk about that in a second too. That was 2014. And then what Obi showed you in terms of rising temperatures, that's in red. You see that upward trajectory? As our temperatures have risen on our reefs, we've seen repeated bleaching events, more than 7 of them since 1985. And the most recent of which in 2013 was the hottest, the strongest, and the longest, and the most devastating that we've seen in the history of studying our coral reefs. Corals have a threshold. It's about 30 degrees, or what's that, 87.1 degrees. When they reach above that threshold, they're not going to do so well. So one of the things I think, being at the Climate Summit, is we often see what's in front of our eyes, but we're often not on reefs. We're not on reefs every day. We may snorkel them every few times. And so sometimes we're missing things happening right before our eyes. And the first event that devastated reefs that kind of went unnoticed was this one. It was a white band disease outbreak. It emerged in St. Croix in 1979, spread across the Caribbean throughout the 80s, and it killed off 95% of our now endangered staghorn and elkhorn corals. If you were on reefs before the devastating impacts in 1979, they looked like this. Staghorn corals everywhere, elkhorn corals at the reef crest, really beautiful. And they started dying across the reef. This would be the equivalent if you walk through a pine forest on your daily walk, and over 5 years, all the pine forest died. You'd notice it. But if it's happening on a reef, you may have missed it. And that's what we did. Now, I'm not as old as starting my reef journey in 1979, thankfully. But for me as a young PhD student, I was studying hybridization between elkhorn and staghorn corals. That was my jam. That's what I loved. But as I got more and more into the field on the reefs, I started asking questions. What was the white band disease pathogen? We don't know. Are they resistant? Are the corals that are still standing today, are they highly resistant to the disease? We don't know. Is the disease highly transmissible? And what that shows is that we have many qualified and amazing scientists, but we have more work to do, and we need to be putting resources behind our science. Okay, so my research lab, we've worked on this, and others have as well. For white band disease, we've identified that it's highly transmissible in the water and through snail pathogens. It's bacterial, and we can treat it with antibiotics. But just this last year, we identified who the primary pathogen is. A strange bacterium called Cystenophyllum. It's a marine bacteria. We still don't know the source. Why did it take us nearly 40 years to identify the pathogen? It's hard, but we need more science. And what you'll see today is I'm going to show you a little bit of data talking about what I think is how we're going to recover our reefs, and that's identifying supercorals. And we started identifying that 15% of the staghorn corals that are remaining are highly resistant to disease. That's going to be one of my hopeful signs of recovery for you today. So what was an important thing that emerged from the white band disease, the devastating impacts of the white band disease? People in Florida like Ken Niedermeyer, who pioneered coral farming, they became the best in the world at being able to propagate corals. Florida is still the best place in the world to grow corals and plant them out on reefs with more than 20 restoration partners. If you've had the pleasure of being on these reefs, many of them in the Keys, what you're seeing is these in situ nurseries where they're growing hundreds of different genotypes or individuals of coral at scale, right? So this is a beautiful, for me, this is like makes my heart, pitter patter. Staghorn coral, thousands of fragments that can be preserved and outplanted on reefs. And this is what we've been doing. We've been farming at scale, willing divers going out on reef, planting thousands of endangered staghorn and elkhorn corals back on our reef, trying to plant them back and recover them back, okay? And we were doing this pretty well. And then a new disease hit. Many of you might know about this impact, but the disease that hit, when it started, it didn't have a name. We hadn't seen it before. We called it something else. We've now called it, unfortunately, stony coral tissue loss disease. What it is it's a disease that is not like white band disease that affects only 2 species. This is a disease that affects 25 or more of our key reef-building corals. There's probably about 35 common species of corals in the Caribbean, so that means that diseases are now impacting almost all of our corals. These are some of our major corals that are infected. In a nutshell, all the major boulder corals infected by stony coral tissue loss disease. All the beautiful brain corals infected by stony coral tissue loss disease. The timeline was that it emerged in Florida, in Miami in 2014, spread through the Keys through 2015, and is now nearly Caribbean-wide. My sites in Panama are unaffected. I think it's one of the last places that has not received the disease. We learned some lessons from white band disease. We knew we could treat them with antibiotic, but you can't treat thousands of corals dying at scale. So what you see in the upper right-hand corner is a practitioner spreading epoxy paste laced with antibiotics, trying to stop the disease on top of that brain coral colony. The second thing we learned is that we needed to preserve genetic diversity. So at the Reef Institute in West Palm Beach here, they have tanks full of multiple species of coral where they're trying to protect and preserve the genetic diversity of these species in quarantine, okay? And we've also learned how to move our in situ reefs where the disease is spreading onto land. So you're seeing land-based nurseries. I think there's at least 5 of them now funded by the state of Florida. This one is Reef Renewals in Ruskin, Florida. It's a large, in the lower corner, it's a large facility where they have up to 10,000 different corals right now. Okay, so that's our second struggle. It's struggle to struggle to struggle with coral reefs. And we also had to learn new tools to outplant slow-growing massive corals. And then the 2023 heat wave hit. So this was the most catastrophic bleaching event. I imagine most of you have lived in Florida. I'd be surprised if you missed this event, right? So in the 2023 heat wave, the heat wave appeared earlier than it ever had before, in July. The Florida Bay temperatures got to 101 degrees. We exceeded our bleaching threshold on June 14th, and we stayed above our coral bleaching threshold for 5 months. The previous bleaching record, we count these things in degree heating weeks. That's how long our corals have been cooking above their bleaching threshold. We did 22 weeks. The previous bleaching event only hit 8 weeks. Okay, so as you would expect, the coral started dying. Okay, the nurseries, the in situ nurseries, people were pulling out the corals from the in situ nurseries and onshoring them in any facility that they could to control the temperature. And many of those corals have not gone back on reefs, okay? And a lot of our corals died in place. So this is the results of a science paper that was published, I think this year, about 2 months ago. And it's documenting the demise of corals that we had outplanted over the years. This is showing you the scale of the heat, and this is showing you the scale of the loss. For me, this is pretty incredible. This is just staghorn and elkhorn coral, the acroporid corals, but across the contingent of groups, they were monitoring 50,000 different corals that were either wild or had been outplanted over the years. And if you look at the red bars at the bottom, moving up the keys, that is the mortality rate, okay? Dry tortugas, complete loss. Lower keys, complete loss, nearly 97%. Middle keys and upper keys, a few survivors. Biscayne Bay, better. Broward County, Miami, better. But here's the thing. The corals survived in the upper portions of the range, not because they were more resilient, more resistant, and they weren't super corals. It wasn't as hot, okay? So it served as a refugia. But in effect, we lost our elkhorn and staghorn corals that we'd been trying to recover for the past 20 years. So I'm sounding like Michael yesterday. I'm like really dour, and he's funnier than I am, so I don't know. I'm just sad, I guess. But here's my hopeful message for you all. I have a few messages. One is that we need to be on the hunt for supercorals. Supercorals has a funny name because many corals can be super, right? But what we're looking for is we're looking for coral species or corals, genotypes, or individuals within individual species that are going to fare better with future climate scenarios, right? Corals are going to survive the heat or going to survive the disease. And it turns out that scientists like myself have been unlocking the secrets of disease resistance and thermal tolerance over the years, mostly through National Science Foundation funding, okay? And so we published this paper in Science 2 years ago where we basically unlocked the code for what makes the staghorn coral disease resistant. Okay, and here's the good news. The way you do this is you take the restoration farmers' genotypes, you challenge them, and you identify the disease-resistant ones. You then apply some fancy genome sequencing to get the genetic variation. And in our surveys, we still get 15 to 20% of our staghorn corals as disease-resistant in the field. We see that, okay? But we can now use and identify 10 genes that explain that disease resistance. This is the only data slide I'm showing today that's mine. The bottom is something called a polygenic score. That's genetics, fancy genetics. The y-axis is disease resistance, okay? Our technique allows us to just do a simple genetic survey in these corals and identify, without having to transmit disease to them, without having to challenge them, whether they're going to be resistant or not. 10 genes. And if you want to look for the supercorals on the graph, they're in teal, up in the upper right-hand corner. Okay, so 15% of them are highly resistant. That lets us go to our restoration partners like Coral Restoration Foundation, Moat Reef Renewal. We can basically provide a genetic tool to tell them which of their corals are going to be disease resistant. We can go into wild populations to do that as well. And we can do that, and the next step is thermal tolerance. Okay, so what I'm showing you on the far right is known variation in thermal tolerance. The 2023 bleaching event provided us a unique in situ experiment to look at how our genotypes are going to perform in future climate scenarios. Hopefully we'll not see 2023 again, but those are bleaching patterns within the reef renewal of brain coral genotypes up top and boulder coral genotypes down below. These are corals through the bleaching event. The brown ones are super. The white ones are dying in place and are highly susceptible. And now we need to unlock what the genetics is going to tell us about these corals so that we can identify the next generation of thermal tolerant supercorals. Okay, so this is my message to leave you with today. We know in Florida, nowhere else can we restore reefs at scale. We have the partners, they're numerous. We can restore reefs at an industrial scale. We have the science nationally and in Florida itself to basically identify the next generation of supercorals. What we lack is the investment. This has been a standard kind of refrain at this entire meeting, right? But I wanted to dig around and give you a scale of what we could be doing, okay? So I pulled some stats. They're my stats as a geneticist, not as an economist, so full disclosure there. We spend about $50 billion, or we get out about $50 billion out of our US forests, right? That's what we're extracting in that resource. And we're treating them like a resource, and we're reinvesting back $8.9 billion. That's a 17% reinvestment in our forests, okay? That's good. That's what you would invest in a healthy business, right? So then let's look at what we're investing in our Florida reefs. $8.6 billion economy, restoration efforts, things like iconic reefs, other things over the year. We're probably putting about $40 million invested back into our reefs in direct restoration. That's 0.5% reinvestment back in our reefs. So my call today is, if we want to tip our scales back in favor of our reefs and expect to have living coral back again, let's match the challenge that's facing our coral reefs with the investment it needs. Let's empower our coral farmers and our scientists, put money back into it, because we know we can do it. We just need the will to do it. That's my pitch. Thank you.